Today, we are taking on a massive mission, charting the secret command structure of the human body.
And we're not talking about nerve impulses here.
No, forget the electrical stuff. We're talking about the sheer silent power of chemical communication. We are peeling back the curtain on the endocrine system.
It's a great way to think about it. If the brain is your central processing unit, then hormones are this meticulously regulated fiber optic network. They coordinate absolutely everything that keeps you alive.
Everything.
They maintain that moment-to-moment stability, what we call homeostasis. But at the same time, they're guiding the huge programmed events of your life, things like growth, reproduction, even aging.
And the name itself tells the story, doesn't it? Hormone comes from the ancient Greek word hormein,
Which means to stir up.
Exactly, to stir up. These molecules stir up action far, far from where they're actually made. We've pulled together a really substantial stack of research for this, some dense endocrinology and pharmacology texts.
And our focus isn't just on the hormones themselves.
No, it's on the true chief executive of this whole chemical network, the pituitary gland.
So our mission today is to give you this high-level but really thoroughly detailed understanding of how the pituitary, specifically that highly active anterior lobe, controls your core survival and reproductive systems.
We need to unpack the core systems, right? And understand the language they use to talk to cells.
And crucially, explore the cutting-edge therapeutic strategies. Because modern medicine, it doesn't just replace these hormones anymore. It actually exploits their own regulatory mechanisms to get these, profound results.
So you're about to master the master gland.
That's the goal.
Okay, let's start by zooming out a bit. Let's look at the sheer scope of this system. When we talk about hormones, we're really talking about regulation that touches every single part of your existence.
It's true. The material we're diving into breaks the body's control systems down into 7 distinct pillars.
7 pillars. And these pillars, they basically define life itself.
They do. The first is the availability of fuel. This is this ongoing negotiation between energy storage and access.
And that's primarily managed by that team of insulin, glucagon, and
interestingly, growth hormone, which affects glucose availability in a very specific way, we'll definitely have to discuss later.
Right. Okay, so pillar 2 is the engine speed, your metabolic rate.
It's your internal thermostat, yeah.
Ah.
And that's almost entirely governed by the thyroid hormones, which are triiodothyronine, or T3 and thyroxine, T4.
Then during development, we've got pillar 3, somatic growth.
The main driver here is, of course, growth hormone, but it's so critical to remember its main executioners.
The insulin-like growth factors.
The IGFs.
Exactly. Then you have the beautiful complexity of sex and reproduction, which is pillar 4. That's all orchestrated by the gonadotropins from the anterior pituitary, FSH and LH, and then the resulting sex steroids like androgens, estrogens.
And progestins. Okay, so moving into stability, pillar 5 is maintaining circulating volume, which is just crucial for regulating blood pressure and fluid balance. The key players there are aldosterone, a mineralocorticoid, and the antidiuretic hormone, or ADH.
Also called vasopressin.
Also called vasopressin, yes.
Pillar 6 sounds essential for, just survival, adaptation to stress.
Oh, absolutely. Both acute and chronic stress responses are coordinated by the glucocorticoids, like cortisol, and that immediate fight-or-flight signal, adrenaline.
And finally, the 7th pillar, the meticulous maintenance of calcium balance. You need that for everything from nerve conduction to bone density.
And that's managed by a trio: parathormone, or PTH, calcitonin, and the active form of vitamin D. It's a huge cast of characters.
A massive cast, but they all communicate through these chemical messages.
And these powerful regulators, they're produced by a collection of ductless glands, the endocrine ensemble. We're focusing on the pituitary today, but the system also includes the thyroid, parathyroid, the pancreas.
Specifically, the islets of Langerhans in the pancreas.
Right. And the adrenals, both the cortex and medulla, and the gonads. And every single one of them is under the ultimate critical control of a single structure.
And that structure is the hypothalamus. I think it's really easy to mistakenly call the hypothalamus a gland.
People do all the time.
But the sources are so clear on this. It is part of the central nervous system. It's the highest neural input that converts electrical signals from the brain into chemical instructions for the pituitary.
And that conversion process involves producing a handful of these incredibly potent releasing and inhibitory hormones. These are the ultimate specific instructions that tell the anterior pituitary what to do.
And they have very different chemical structures, which is key.
It is.
So let's detail the 6 keys because they're really the root of the entire system. We can start with TRH, thyrotropin releasing hormone. Chemically, it's tiny.
just a tripeptide, 3 amino acids.
Then you have CRH, corticotropin-releasing hormone, which is a much larger peptide of 41 amino acids.
And then the reproductive master switch, GRH, gonadotropin-releasing hormone.
That's a decapeptide, right? 10 amino acids.
10 amino acids, yes. And we'll see that its specific secretion pattern, not just its presence, is the absolute key to unlocking the entire reproductive cycle.
And what's fascinating about GnRH is that it controls both FSH and LH. We'll definitely have to get into that pulse theory later on.
Oh, we will.
But let's look at the exception to the rule in this group. The primary regulator for prolactin, PRIH.
Prolactin release inhibitory hormone. And that is actually dopamine.
This is a major structural anomaly within the system, isn't it?
It really is. Dopamine's not a peptide. It's a small, modified amino acid, a neuroamine. Its role as the dominant inhibitor for prolactin is this core regulatory mechanism that we exploit heavily in pharmacology.
And finally, the two factors for growth hormone. GHRH, the releasing hormone.
Which is a 40 or 44 amino acid peptide.
And its opponent, somatostatin, or GH-release inhibitory hormone, a 14 amino acid peptide.
And understanding the chemistry here, tripeptide, decapeptide, dopamine, it's crucial because it dictates how rapidly these signals can be produced, transported, and then broken down.
And all of them are delivered to the anterior pituitary through a very specialized route.
The hypothalamo-hypophyseal portal system. It's not just general blood circulation, it's a dedicated chemical highway.
A highway that ensures a rapid, high concentration of these signals arrives exactly where it needs to control that master switch.
Precisely.
Okay, so let's unpack this core problem of communication. You have an instruction, a hormone, floating around in your bloodstream. How does that instruction actually bridge the gap and fundamentally change a cell's behavior? Force it to make a protein or change its metabolism.
This is where we shift from that macro view of the glands to the micro view of molecular action. The body uses 3 primary communication pathways.
And the specific mechanism is totally determined by the hormone's chemical nature.
Exactly. Specifically, whether it can cross the cell's lipid membrane or not.
So let's start with the fastest communication. The stuff you need for quick adjustments, like a stress response.
Right. This is the pathway used by the large peptide hormones, things like adrenaline, TSH, ACTH, and glucagon.
And since they're water-soluble, they just can't cross that lipid barrier. So their site of action is at the cell membrane.
That's it. They act as the first messenger. They bind to a receptor on the outside of the cell, and that instantly activates an enzyme system on the inside, generating a second messenger to relay the instruction.
And the first main system they use is the KMP system.
The KMT system, used by hormones like TSH, PTH, adrenaline acting on beta receptors, a whole host of them.
So when the hormone binds, it activates what's called a G-protein-coupled receptor, or a GPCR.
Right, and this G-protein then turns on the enzyme adenylyl cyclase, which catalyzes the formation of cyclic AMP-KMP from ATP.
So the concentration of this internal messenger, KMP, it changes very, very rapidly.
And what does that change in KMP actually do?
It acts like a dimmer switch, right?
A perfect analogy. Elevated KMP activates protein kinase A, or PKA. And PKA is this major regulatory enzyme that then goes on to phosphorylate or add a phosphate group to countless other enzymes and proteins inside the cell.
This is a cascade effect. One hormone molecule can trigger this enormous internal change.
An enormous change. It rapidly regulates enzyme activity, permeability, secretion. It is the ultimate amplification system.
That makes perfect sense for rapid regulation. What about the second major pathway at the membrane?
That would be the IPD degeneration system. This involves a different type of G protein and a different enzyme. When hormones like vasopressin on its V1R receptors or oxytocin burned, the G protein activates phospholipase C.
And phospholipase C, that's what breaks down a component of the membrane itself, PIP22-ups.
Precisely. And that breakdown gives you 2 second messengers, inositol trisphosphate, or IP3 and diacylglycerol, which is D.
Okay, so 2 for 1.
2 for 1. And they do different things. IP3 does this one key job. It causes the rapid release of stored calcium ions from internal organelles, mostly the endoplasmic reticulum. Digase stays in the membrane, and it activates another enzyme, protein kinase C, or PKC.
So now we have 2 different protein kinases being activated, and we've introduced a third messenger in calcium. Why the extra complexity? How does that help the cell?
Well, calcium is an incredibly versatile signal. It can bind to other regulatory proteins like calmodulin, which then modulates a whole new set of enzymes. Meanwhile, PKC is phosphorylating its own set of target proteins.
So it gives the cell more options.
It gives the cell a parallel, distinct set of regulatory levers from the CAMP system. You see this IPDJ pathway a lot with things like cellular contraction, secretion, and membrane permeability changes.
And then there's the third membrane pathway, which is unique. This is the one for some of the most critical anabolic hormones. Direct transmembrane activation of tyrosine protein kinase.
The method of action for insulin, growth hormone, and prolactin.
And this is more of a direct engagement.
It is. When the hormone, let's say growth hormone, binds, it physically forces two receptors to come together to dimerize. And this dimerization activates an enzyme, tyrosine kinase, that is either built right into the receptor or is very tightly associated with it.
Like the JAK-STAT pathway.
Exactly, which we'll see with GH and prolactin. The moment that kinase is activated, it starts a phosphorylation cascade.
And this one is less about rapid enzyme changes and more about the long game.
Very much so. It's more about regulating gene expression and promoting growth. The phosphorylation leads to the activation of transcription factors, like STATs, which then migrate to the nucleus and kick off the synthesis of new proteins. This is the pathway for long-term metabolic and growth-promoting effects.
Okay, that covers the peptides that stay outside. Now we move inside the cell, where the hormones are fat-soluble. This is the slower but maybe more profound communication system at cytoplasmic receptors.
This pathway is characteristic of all the major steroid hormones. So we're talking about glucocorticoids, mineralocorticoids, the sex steroids,
Androgens, estrogens, progestins.
and calcitriol, which is active vitamin D. Because they're lipid-soluble, they just diffuse right across the cell membrane. No receptor or transporter needed on the outside.
So once they're inside, they find their specific receptor just floating in the cytoplasm. What happens then?
The binding causes the receptor to shed its chaperone proteins and undergo this crucial conformational change, which exposes a DNA-binding domain. This whole complex, the hormone plus its receptor, then moves directly into the nucleus,
Acting like a specialized key. It finds its specific DNA sequence.
which is known as a hormone response element, or HRE. By binding there, it directly modulates the rate of transcription. It initiates DNA-mediated mRNA synthesis, which ultimately dictates the long-term synthesis of new proteins.
So this is a complete cellular overhaul. It changes the cell's entire machinery.
Absolutely. And it's a slow process. It can take hours or even days, but the effects are sustained and fundamental.
And finally, the third site of action is similar, but distinct. At nuclear receptors, this is the domain of the thyroid hormones, T3-threonate and T4-4.
The thyroid hormones are also lipophilic, so they can penetrate the cell. But unlike steroids, they mostly pass right through the cytoplasm and combine with a receptor that's already sitting directly inside the nucleus.
But the result is the same.
The end result is the same: activation of specific genes to alter DNA, RNA-mediated protein synthesis.
So what really stands out here is that the body matches the regulatory task to the molecular communication method. Quick survival, like with adrenaline, uses that rapid KMP cascade.
And long-term structural change, like with steroids or thyroid hormone, uses direct gene regulation in the nucleus. It's a highly efficient system. You don't want a stress hormone causing a several-day metabolic shift, and you don't want growth to be regulated minute by minute.
We have established the language. Now let's focus on the speaker of that language, the anterior pituitary, the adenohypophysis. It really does earn the title, Master Endocrine Gland.
It absolutely does. And as we just discussed, all 6 of its major hormones are peptides.
Which means they all operate on those extracellular membrane-bound receptors.
Right. And this gland is complex internally. The cells that secrete these 6 hormones are differentiated by their staining properties.
We can break them down into 2 main groups. First, you've got the acidophil cells.
The acidophil group includes the somatotropes, which produce growth hormone, or GH, and the lactotropes, which secrete prolactin, PRL. And they're structurally related. They often share the same signaling pathways.
Then you have the basophil cells, which are responsible for the regulatory hormones.
And that group is larger. It includes the thyrotropes making TSH, the corticotropes, lipotropes making ACTH, and the gonadotropes, which produce both FSH and LH. It's an incredibly crowded production floor.
5 distinct cell types pumping out 6 major hormones. Now, we keep coming back to this idea of hypothalamic control. How does the body make sure the anterior pituitary doesn't just run wild and release everything constantly?
It's under constant, strict supervision. 2 mechanisms. First is the input. Those 6 releasing and inhibitory hormones we talked about are delivered via the hypothalamo-hypophyseal portal system.
That dedicated highway.
That highway, the capillary network ensures the signal concentration is high and immediate. So the hypothalamus dictates the baseline.
Okay, so that's mechanism one.
And the second mechanism is the crucial feedback inhibition. Once the pituitary hormones act on their target glands, like the adrenals, thyroid, or gonads, the hormones that those glands produce,
Cortisol, T4, testosterone.
they loop back. They inhibit the pituitary and often the hypothalamus itself, basically telling the system, mission accomplished, you can stand down.
And from a therapeutic standpoint, this is where all the action is. One key distinction really jumps out from this material. We don't always need to replace the missing pituitary hormone, do we?
No, not at all. Often we need to fine-tune its release. TSH and ACTH are mainly diagnostic tools. But for growth hormone and the gonadotropins, we've advanced to a stage where we are chemically stimulating or, more often, chemically inhibiting their secretion by manipulating these hypothalamic controls and feedback loops.
All right, let's dedicate some serious time to growth hormone. This is a 191 amino acid single-chain peptide, and it's really the central engine of somatic growth.
Its role in childhood is obvious, right? It's for attaining adult stature. GH ensures the growth of virtually all organs.
By inducing both hyperplasia, which is more cells,
And hypertrophy, bigger cells.
in almost every organ, with the notable exceptions of the brain and the eye.
Right, which largely develop on their own timeline. But I think a lot of people underestimate its lifelong metabolic effects. It's not just for kids.
Not at all.
GH is profoundly anabolic. It promotes a positive nitrogen balance.
Meaning it increases the uptake of amino acids and protein synthesis, helping you build lean body mass.
Right, and at the same time, it's a powerful catabolic agent for fat. It promotes fat utilization lipolysis, which in turn helps to spare carbohydrates and protein.
Okay, but if it's promoting fat breakdown, that sounds like it would tend to raise blood sugar.
It does. GH is diabetogenic. It raises blood glucose by reducing glucose uptake in muscle tissue and enhancing glucose output from the liver.
Gluconeogenesis and glycogenolysis.
Exactly. And this is a critical dual metabolic role, and it brings us to the most fascinating part of its action, the mediator connection.
Okay, here's where it gets really interesting. You've emphasized that GH's primary growth and anabolic actions are largely indirect. It uses a middleman.
It uses the somatomidins, principally insulin-like growth factor 1, IGF-1. Think of GH as the long-distance signal, and IGF-1 as the local messenger that actually executes the growth instructions.
So GH acts on the liver, and the liver becomes the major source of circulating IGF-1, which then acts on distant tissues.
And you also get locally produced IGF-1 acting in a paracrine manner, meaning it influences neighboring cells right there at the growth plate or in muscle tissue. But this leads back to that interesting paradox we touched on.
Yes, the paradox. IGF-1 being insulin-like, it promotes glucose uptake and lipogenesis. Those are anti-diabetic actions.
But the mother hormone, GH, acts directly to promote lipolysis and raise blood sugar.
So if you have too much GH, you get simultaneous signaling for massive growth through IGF-1 and profound metabolic stress through GH's direct actions.
It accentuates the metabolic derangement you see in conditions like diabetes because GH is actively fighting insulin's efforts to lower blood sugar and store fat. It's a constant tug of war.
And this tight regulation is managed by that GHRH and somatostatin feedback loop.
Both hypothalamic peptides act on G-protein-coupled receptors on the somatotropes in the pituitary, modulating CAMP, but the crucial negative feedback comes from the product of the pathway.
From IGF-1 itself.
Right. IGF-1 tells the hypothalamus and pituitary to throttle back GH release.
Let's talk therapy. The advent of recombinant human GH, somatropin, it completely revolutionized the treatment of pituitary dwarfism.
Oh, it was life-changing. For true GH deficiency, a daily subcutaneous injection of somatropin is a highly successful therapy. You can often achieve near normal adult height.
Provided treatment begins early and continues until the growth plates fuse around age 20.
And it's also approved for more specific syndromes that cause short stature, things like Turner syndrome or chronic kidney failure in children. But its use in constitutional short stature is very controversial.
That's where the ethical debate really comes in. We're talking about treating constitutional short stature where a child is simply genetically small but otherwise totally normal.
And it involves years of very, very costly medication, injections, regular monitoring, just to gain maybe an inch or two for social or cosmetic reasons.
It forces clinicians and parents to weigh the long-term cost and the potential risks against this, social pressure of conformity. It's a debate between medical necessity and enhancement.
And the sources are very clear that GH isn't just a childhood hormone. Its use in adults with GH deficiency shows pretty substantial results.
For adults, GH replacement therapy can significantly increase lean body mass, dramatically decrease body fat, and generally improve energy, mood, and work capacity.
And it's also used in catabolic states like AIDS-related wasting or severe burns, which really demonstrates its potent protein-sparing anabolic effect during profound stress.
But we have to mention the risks. Its non-regulated use, especially in the anti-aging sphere or by athletes, is banned.
It's on all the dope testing lists, and the adverse effects are serious. Besides the risk of glucose intolerance, you can see hypothyroidism and salt and water retention, which leads to edema.
Which brings us to the inhibitory side of the equation. For conditions like acromegaly or gigantism, where you have excess GH, you have to shut the system down. And the initial molecule is somatostatin.
But somatostatin is notoriously nonspecific, right? It's a shotgun approach.
It is. It inhibits GH, but also prolactin, TSH, and major digestive hormones like insulin and glucagon. And its half-life of 2 to 3 minutes also makes it clinically impractical for any kind of long-term use.
That inefficiency led directly to the development of synthetic analogs like octreotide.
And octreotide is an engineered success. It's about 40 times more potent at suppressing GH and has a much more manageable half-life of around 90 minutes. It's the preferred agent for acromegaly.
And for controlling those severe secretory diarrheas caused by certain tumors. But because it still hits multiple receptors, you see things like gallstones and GI side effects as common long-term issues.
And then we have the specific molecular weapon, pegvisomant. This is a totally different approach to inhibition.
What's fascinating here is that pegvisomant doesn't try to stop the pituitary from making GH, it stops the GH from working. It's a mutant GH molecule.
Complexed with polyethylene glycol.
Right, and it acts as a pure GH receptor antagonist. It binds to the receptor, but instead of causing that dimerization and activation of the JAK-STAT pathway, it just blocks the binding site. It prevents any signal transduction.
So it's the perfect blockade.
Highly specific, and it's a vital therapeutic option for acromegaly patients who don't respond to octreotide.
Okay, moving from growth hormone, let's tackle its chemical cousin, prolactin, a 199 amino acid peptide. Its primary role in human physiology is really tightly focused on reproduction and feeding the young.
Prolactin's name tells you its function, right? Promotion of lactation. But its actions are stepwise. During pregnancy, it works alongside high levels of estrogen and progesterone to cause the essential physical growth and development of the mammary tissue.
But the actual milk secretion, the lactation, that doesn't begin until after the baby is born. Why the delay?
Because those high estrogen and progesterone levels during pregnancy are actually inhibitory to the final stages of milk synthesis.
Ah, okay.
After parturition, when those steroid hormones rapidly withdraw, prolactin is finally able to induce the full biochemical machinery needed for making milk proteins and lactose. The suckling stimulus then causes bursts of prolactin release to sustain lactation.
And here is where prolactin plays its critical role in fertility control. The suppression of the hypothalamic pituitary gonadal axis.
Right. High continuous levels of prolactin sustained by frequent feeding, they inhibit the release of GnRH from the hypothalamus.
And we already said GnRH is the master reproductive switch.
So by suppressing GnRH, prolactin suppresses FSH and LH, leading to lactational amenorrhea, the temporary end of the menstrual cycle, and resulting infertility. It's a biologically programmed mechanism to space out pregnancies.
This brings us back to the regulatory system and the unique position of dopamine. If dopamine is the inhibitor, how does the body turn the system on to start lactation? And what happens when that balance fails?
This is a truly beautiful exception to the rule. Prolactin is under dominant inhibitory control. The hypothalamus is constantly pumping out PRAH, which is dopamine, acting on the TEX-D receptors of the lactotropes.
So it's always being suppressed.
Always. Neural input from suckling or stress or even TRH inhibits the dopamine release. That allows the prolactin switch to flip on. It's released in the absence of inhibition.
So it's like a car parked with the emergency brake constantly on. You release the brake, the dopamine inhibition, to make it move, to get prolactin release.
Exactly. And this is why dopamine antagonists, like many common neuroleptic drugs used in psychiatry, or the antiemetic metoclopramide cause hyperprolactinemia as a common side effect. They block that TEX-D receptor, mimicking a complete lack of inhibition.
And hyperprolactinemia is the primary pathology we treat. It causes galactorrhea, amenorrhea, and infertility in women.
And in men, depressed fertility and loss of libido, often due to a prolactin-secreting tumor, a prolactinoma.
So since we never need to use prolactin therapeutically, the entire focus is on inhibition.
Specifically using dopamine agonists. The older drug is bromocriptine, which is a synthetic ergot derivative.
And bromocriptine is a potent TEX2 agonist. It effectively suppresses prolactin release and can even shrink prolactinomas. It also has a secondary use in acromegaly.
It does, but bromocriptine is plagued by side effects. Nausea, vomiting, postural hypotension are very, very frequent, and they often limit the maximum tolerated dose. Plus, its short half-life requires daily dosing.
The pharmacology evolved dramatically with the introduction of cabergoline. This is the truly superior drug.
Cabergoline solved all those problems. It's newer, much more potent, and far more selective for the pituitary TEX2 receptors. But most importantly, it has an incredible half-life, over 60 hours.
Which means patients only need to take it once or twice a week.
That reduction in dosing frequency and the lower incidence of side effects, especially that severe nausea, is a massive quality of life improvement.
So cabergoline is now universally the first choice drug for hyperprolactinemia.
It is. It often causes dramatic regression of prolactinomas and restores fertility. It's a perfect example of optimized pharmacology, improving patient compliance and efficacy by targeting a very specific inhibitory mechanism.
All right, next let's look at the duo essential for reproduction. The gonadotropins, follicle-stimulating hormone, FSH, and luteinizing hormone, LH.
These are complex glycoprotein hormones, and their structure is fascinatingly similar. They share a common, interchangeable alpha chain 92 amino acids long.
But their difference, which determines their specific function, lies entirely in the beta chain.
Right. 111 amino acids for FSH and 121 for LH. They act on G protein-coupled receptors in the ovaries and testes, mainly increasing CAM-MP and stimulating the conversion of cholesterol to pregnenolone, the starting point for all sex steroids.
Their coordinated action is just a beautiful piece of biological engineering.
In females, FSH starts the process. It induces the growth of ovarian follicles and stimulates estrogen secretion. Later, LH is responsible for the dramatic events, triggering preovulatory swelling, causing ovulation itself,
And then stimulating the luteinization of that ruptured follicle to form the corpus luteum.
which sustains progesterone secretion. In males, FSH supports spermatogenesis, and LH, often called ICSH in men, stimulates testosterone secretion by the interstitial cells.
Now, we established that a single hypothalamic factor, GnRH, releases both FSH and LH. This raises a really important question. If you only have one control switch, how does the body achieve those radically different secretion patterns? Favoring FSH early on and then triggering that massive LH surge mid-cycle.
And the answer is one of the biggest insights from modern endocrinology, the GnRH pulse theory.
It's not continuous.
Not at all. GnRH is secreted in discrete, rapid pulses every 60 to 90 minutes, and the pituitary is able to interpret the frequency and the amplitude of these pulses to modulate its output.
So the same receptor responds differently based on the rhythm.
Precisely. During the follicular phase, you see high-frequency, low-amplitude pulses maybe every 60 minutes. That rhythm differentially upregulates the genes for FSH synthesis and release.
And later in the cycle.
Later in the luteal phase, the frequency slows down. You get lower frequency, higher amplitude pulses maybe every 4 hours. And that rhythm favors LH synthesis and secretion. The pulse pattern effectively programs the pituitary.
That is so complex. It's not just the presence of the signal. It's the rhythm of the delivery.
And the system also uses other factors like inhibin, which is secreted by the gonads, to selectively inhibit FSH release, adding another layer of fine-tuning.
In the clinical world, exogenous gonadotropins are essential for treating infertility.
Either when a patient's own pituitary production is deficient, or, more commonly, for controlled ovarian stimulation for things like IVF.
Let's review the preparations. We use menotropins, which are purified from the urine of postmenopausal women.
And that contains equal amounts of FSH and LH. You also have urofolitropin, which is pure FSH.
And pure FSH is often preferred in women with polycystic ovarian syndrome, or PCOS, because they might already have high endogenous LH levels.
Right, you want to balance the ratio. And the critical final piece is human chorionic gonadotropin, or HCG.
HCG is from the placenta, not the pituitary, but its chemical structure is similar enough that its action is functionally indistinguishable from LH.
So in a typical infertility protocol, you'd give menotropins or FSH daily for about 10 days to mature multiple follicles. Then, to trigger the final event, you give a single large dose of HCG to mimic the LH surge.
And ovulation occurs predictably 24 to 48 hours later, allowing for precise egg retrieval or timed intercourse.
But to make that process predictable, you run into the challenge of the body's natural cycle. You don't want the patient's own hypothalamus to suddenly trigger a surge and ruin the timing.
Correct. Modern practice requires concurrent suppression of the patient's natural FSH and LH secretion. And this is where the truly counterintuitive pharmacology comes in.
Using continuous GnRH agonists to achieve inhibition, functionally known as chemical castration.
This concept is so important, using continuous stimulation to cause a profound shutdown.
The superactive GnRH agonists like leuprolide and goserelin are incredibly potent. Because they're delivered continuously, not in pulses, they initially stimulate a brief flare-up of FSH and LH.
But after a week or two,
After 1 to 2 weeks, this continuous non-pulsatile signal just overwhelms the pituitary. The GnRH receptors are desensitized and profoundly downregulated.
so the system effectively loses its ability to respond.
It shuts down completely. FSH and LH secretion ceases, and as a result, estrogen and testosterone levels fall dramatically to postmenopausal or castration levels. It's a reversible pharmacological shutdown.
And it's used for conditions that are hormone sensitive, like prostatic carcinoma, endometriosis, uterine fibroids, or precocious puberty.
And of course, suppressing the endogenous cycle before induced ovulation starts in IVF.
The alternative method is the GnRH antagonists, like ganirelix and cetrorelix. These are just pure competitive blockers.
And the advantage there is clear. They achieve quick GN suppression by physically binding to the receptor and blocking the GnRH signal. They bypass that initial, often undesirable flare-up phase you see with the agonists.
Right, so they're started later in the IVF cycle, and they're generally preferred because they offer a shorter, quicker suppression.
Which carries a lower risk of severe complications, particularly ovarian hyperstimulation syndrome, or OHSS.
And speaking of risks, we have to acknowledge the seriousness of OHSS, where the ovaries become hyperstimulated. It can lead to major complications.
And the ever-present risk of multiple pregnancy from aggressive stimulation. But for long-term use of the GnRH agonists, when you're treating endometriosis for many months, the sources really stress the severe risk of developing osteoporosis due to that prolonged, profound estrogen deprivation.
Okay, we've covered the therapeutic powerhouses. Now let's briefly turn to the two remaining anterior pituitary hormones, TSH and ACTH. In clinical practice, their roles are mainly diagnostic.
Let's start with thyroid-stimulating hormone, TSH, or thyrotropin. TSH is responsible for stimulating every single step of thyroid hormone synthesis and secretion.
Its actions are broad. It causes growth and hypertrophy of the thyroid follicles, it increases blood supply, and critically, it promotes the uptake of iodide from the circulation into the thyroid cells.
By enhancing the sodium iodide symporter, or NIS, it uses the GPCR-AMP system to accelerate organification, coupling, and the final release of T4 and T3
Regulation is simple. Hypothalamic TRH releases it, and high levels of T3 and 2,4-100 provide negative feedback.
Primarily at the pituitary level. And the clinical takeaway is that thyroxine is the preferred treatment for hypothyroidism, not TSH itself.
So why do we care so much about TSH levels?
Because TSH is the ultimate diagnostic tool. If a patient is hypothyroid, we check TSH. If TSH is sky high, we know the patient had primary thyroid disease.
Meaning the thyroid gland itself is failing and the pituitary is just screaming for action?
Exactly. But if the patient is hypothyroid and TSH is inappropriately low or normal, we know the problem is upstream. Secondary disease from pituitary or hypothalamic dysfunction. TSH levels differentiate the origin of the disorder.
And finally, we turn to adrenocorticotropic hormone, ACTH, or corticotropin. This is a 39 amino acid peptide derived from a huge precursor molecule.
ACTH is the body's primary response to stress. It manages the output of the adrenal cortex. It promotes steroidogenesis by rapidly stimulating CAMP formation, which accelerates the conversion of cholesterol to pregnenolone.
And that conversion is the crucial rate-limiting step for almost all glucocorticoids, mineralocorticoids, and adrenal androgens. So ACTH effectively turns on the entire adrenal steroid assembly line.
And it also has a trophic or growth-promoting influence on the adrenal cortex, maintaining its structure. Regulation is governed by CRH, and ACTH is famous for its circadian rhythm.
Right. It peaks first thing in the morning, preparing you for the day, and it's lowest around midnight.
But the body has a failsafe during a crisis. Stressful stimuli completely override the normal rhythm in the feedback loops.
So during trauma or surgery or profound fear.
The neural inputs converge on the hypothalamus, causing a massive release of CRH. And that signal is so powerful, it completely overpowers the negative feedback from high cortisol levels. The system demands cortisol for survival, no matter what's already circulating.
And similar to TSH, ACTH itself is rarely used therapeutically. We prefer synthetic corticosteroids.
Right. It is used diagnostically to assess the adrenal axis, but the most common pathology related to ACTH that clinicians deal with daily is iatrogenic suppression.
Meaning suppression caused by medical treatment.
Exactly. Patients treated with exogenous glucocorticoids for other diseases often suppress their own pituitary ACTH production. This leaves the adrenal cortex atrophied, which requires a very careful, gradual weaning off the steroids to allow the axis to recover. Otherwise, the patient risks adrenal crisis.
We have spent a significant amount of time traversing the very complex landscape of the anterior pituitary. It's a tiny gland, but its 6 peptides manage everything from metabolism and growth to fertility and stress response.
What really stands out is just the elegance of the regulatory mechanisms. The system relies on these finely tuned chemical signals like the hyperthalamic factors, but also on these deep layered feedback loops.
We saw the paradox in the GH and IGF-1 axis.
The counterintuitive nature of dopamine as the dominant inhibitor for prolactin and the complex rhythm required by the GnRH pulse theory to orchestrate reproduction. It's incredible.
And the takeaway for you, the learner, is how sophisticated modern pharmacology has become by exploiting these exact mechanisms.
Yeah.
We don't just replace hormones anymore.
No. We use continuous GnRH agonists to deliberately cause receptor desensitization and achieve a state of pharmacological shutdown. Or we use highly selective, long-acting antagonists like cabergoline to provide precise, low-frequency control.
You now have a comprehensive understanding of these master switches. Well, let's leave you with a provocative thought. We already noted the controversy surrounding growth hormone use for constitutional short stature and anti-aging.
Right.
If we connect this to the bigger picture, how much of future medicine will focus not on fixing pathology, but on chemically tweaking these foundational pituitary systems, GH, ACTH, FSH, LH, purely for human optimization, enhancement, or cosmetic effects?
We are increasingly capable of chemically overriding natural biological limits. What ethical and societal challenges will arise as that pharmaceutical sophistication becomes more accessible? You now have the knowledge to critically evaluate that debate.
A perfect foundation for critical thought.
Yeah.
Thank you for diving deep with us. We'll see you next time.